old physicist joke, right? So they say, hey, let's assume a spherical cow. I used to hate biology like everybody else in school.
It's an
I
either
hate
or.
biology, so I go for math.
Yeah.
I can't do math, so I'll go into medicine or biology. Usually share in the first class in my course on systems biology. Right? This is called Can a Biologist Fix a Radio? Compound called surfactant in the International Space Station microbes. Heavily used in cosmetics. Deep sea, gut, International Space Station. How do these microbes interact with each other? You literally have to, everything has to be done by reverse engineering.
Hi, this is Amrit. We are at IIT Madras, my alma mater, and India's top university for people who like to build. We are here to meet some builders, ask them, what are you building? What does it take to build? And what makes IIT Madras the best place to build?
Today on the Best Place to Build podcast, we have with us Professor Karthik Raman, a professor in the Department of Data Science and AI in the Wadhwani School of Data Science and AI at IIT Madras. Previously, he was a professor in the Biotech Department. Professor, welcome to the podcast.
Hi.
I also personally know you from our Ask IITM demo days. You've been answering questions when students come in. They have a lot of questions about how IIT works. And I want to just tell you that his answering speed is insane. He'll answer at one question per second. Professor, we had a conversation earlier this week about the work you do, and I've come to realize that there is a lot that I don't understand at all. So, if I may ask, can you give me a basic primer? And I have noted down some words.
Sure.
So maybe you can help me understand this. And without this, everything is going to be OHT for me. So,
Sure.
What is systems biology?
Yeah. So I think, it's because in a sense, biology has become quite, diverse from engineering in a lot of our education, right? So I think that's one of the reasons, and I hope, we'll talk about that and emphasize that at some point of time. But I think it's very important today to be able to straddle both biology and math and so on. And if you look at systems biology, it's essentially like, there are various flavors of computational biology I like to think of, right? So there is, you'll see this term quantitative or computational biology in a lot of scenarios, or you must have heard of the term bioinformatics, right? That's, there are even degrees on bioinformatics and so on. So I like to think of bioinformatics as IT intersection biology, computational biology as computer science intersection biology, and systems biology actually as engineering or chemical or computational engineering intersection biology, right?
Okay.
So that is what I like to think of. But essentially, the whole idea is that we today have the technology, the wherewithal to build complex models of various kinds of systems, right? So the idea is that we can build these complex models to understand more about these systems, right? Because you cannot just, if you look at a lot of traditional biology, there would be, you look at all the Nobel Prizes that were awarded for the last several decades, you will see that they would focus on identifying like one protein, one pathway, and so on. That is absolutely critical. But the thing is that does not give you like a holistic picture of what happens within a cell. And suppose the whole idea is to, so there's Tom Knight who once said, can we tame biology as an engineering discipline? Right? Because in an engineering discipline, you always know, you have a mobile phone, you know how it works, right? You know what are the components, how it works, how it functions. You pull this out, it doesn't work. You put this, you replace this, this starts working, and so on. But in biology, you really cannot do that, right? So you do not have like a full picture of what happens within a cell. Can we try and get close to that in some fashion by building models of these complex systems? So that is the idea of systems biology, where the idea is I want to be able to ultimately manipulate these systems. So I need to know what happens when I touch different parts of the system. So if you see, one of the most popular methods of studying biological systems is by perturbation, right? So you take something...
Professor, sorry, you're moving too fast for me, so I'm going to break you a bit.
Yeah.
I just want to pick up a word that you used. You used the word modeling.
Yes.
What does that mean?
So modeling is just, a mathematical modeling. So I mean, we've all been exposed to models right from school, right? So you've definitely seen a ball and stick model in your chemistry class, right? It shows that, there are these kinds of angles in the methane molecule or something like that. So what you have is that every model essentially tries to mimic certain interesting characteristics of a real world system. A ball and stick model, for instance, scales reasonably, right? It gives you an idea of what is the size of the molecule with respect to the bond. But of course, it's like much bigger than a real molecule actually is, right?
Yeah.
But then you understand what is the angle between the different carbons and hydrogens and so on when you look at the ball and stick model. So every model that way is interested in mimicking a part of reality so that you can understand it better. And what we do is we build models of biological systems. For instance, a classic model that people were really splitting hairs or even trading blows was how does COVID spread? Right. So this is like a very interesting model for people to figure out. And COVID was just notorious there. It was so difficult simply because there's so many more layers. People have been building epidemiological models for maybe centuries, and they all got stumped when confronted with COVID.
Can I just ask you here, do you mean how did COVID spread in the population? Or do you mean
so what is the rate of spread, right?
Itself?
So what is, no. Just what is the rate of spread? Because typically some of these are like higher level models. So you can build these models at different level of abstractions. I can also say, hey, what is the next variant going to emerge? That's going to be far harder, and I don't think anybody has solved that problem. But more likely you're interested in how many beds do I need in the hospital?
Correct.
How much oxygen should I
And I remember
produce?
that we all learned the word, the term R. I think, was it R?
I think BRR, right? So basic reproductive rate or something like that, like R naught, right?
Yeah,
so
R
what
naught,
R
yeah.
naught, right? So what is the rate at which? So if so, that is when you announce something as a pandemic, essentially, right? So when R naught is high, you say it's a pandemic. The rate at which the epidemic is growing is much higher than the rate at which you're treating, or people are getting less infective and so on. And these are like, there is a very famous and simple model that everybody can look up. It's called the SIR model. It's a very simple model. It just super simple. I can explain it in a minute. Just says split the population into susceptible, infectious, and recovered. So all people are potentially susceptible. Some people have gotten infected. Some have recovered. They might again become susceptible or whatever and so on. So just this, right? So how does the rate of, and this is like classic differential equations that you'll write in, like, say, a chemical engineering course or a mechanical engineering course. So ds by dt, di by dt, dr by dt. Set up the equations and you try to predict what is the rate at which the infectious people are increasing, right? And this is what you want to figure out, right? So essentially these models try to answer certain important questions that we want to ask of a biological system.
You're giving this COVID as an example.
As
of
an example,
modeling.
yeah. It's something, in fact, we haven't really worked much on, but it's like a classic problem that I teach in my course on systems biology and so on. Because this is like a nice way to start thinking about how will you model a system. Because every time you're confronted with the modeling challenge, you're going to pick, hey, I'm going to assume that these things don't exist. I'm not going to assume resistance. I'm not going to assume vaccination. I'm going to keep my model simple and then start incrementally building the model, right? So the model is basically a very simplified construct of any real world system trying to answer certain questions. So you may now, at the complete other end, you have these things called digital twins that are becoming really hot today, right? Yeah. So you really want to talk about digital twins. That's essentially like a computer program or a model or something that you have access to on a computer, where you go and tap stuff there. It has to behave exactly like your real world system.
Yeah. And yeah, I want to just say, because one of your lectures I was listening to, you mentioned the spherical cow model, which is take a cow, assume it's a sphere.
So this is the joke, right? So this is like the an old physicist joke, right? So they say, hey, let's assume a spherical cow in a vacuum, ignoring the effects of gravity.
Yeah.
Right. So
so that's the opposite end of digital twin, where
that's the opposite end, right? But I think that's a reasonable place to start, even, right? So as a modeler, you want to start with spherical cows, even. I mean, even though we mock them and so on, because it's like you start fixing certain ideas. You at least start capturing the simplest concepts. And one of my colleagues always gives this example of, looking at when you do your Chandrayaan or Mangalyaan and so on, you ultimately, some of the models assume Earth, Mars, Moon as point objects.
Yeah, as dots.
Right? Yeah. Right? With like a mass, which is very important. Right? So that mass helps you compute the escape velocity and so on. We also have studied that in school. But all these models are actually useful, and there is a very famous quote by a statistician called Box, who just says all models are wrong, but some are useful, right? So that's the whole point, right? So you'll still have wrong models, but which can still give you very interesting or useful insights about real world systems, because we will make potentially atrocious approximations, but you'll still be able to answer some very interesting questions about a real world system.
Understood. But the example we took is of a macro system. I see that your work is more to do with microbiology, right?
Exactly.
So what would be a microbiology example for a modeling problem?
A single cell, right?
Okay.
So how do I manipulate a single cell, right? So for instance, my PhD work was on how do we kill tuberculosis. How do we kill the organism that causes tuberculosis? There's this organism called Mycobacterium tuberculosis. It has 4,000 genes. Which of those 4,000 genes do you go and target with a drug? Okay, so these are some very interesting questions that one tries to answer. So I need to study what these genes are making. So every gene typically makes something known as a protein. And these proteins, essentially, they'll contribute to various things. For example, our hair or our body structure, there are structural proteins that hold these things in place. But inside a microbial cell, there are lots of proteins that catalyze various chemical reactions. So which of these chemical reactions is so essential for the cell that if you go and target it with a drug, the cell ceases to function?
Understood.
So these are the questions that we try to answer, right? So how do you pick the right proteins to target? The problem is certain proteins, if you target, there'll be other side effects, right? You may not be able to uniquely target that protein. By targeting that protein, you might end up hitting some human proteins, or you might end up hitting some other proteins of other beneficial bacteria that live in the body, right? We will talk about the gut microbiome later on, which is one of my favorite topics and a lot of what our lab works on.
Yeah.
But you want to be very selective in how you go and hit these systems, right? And for which you need a systems perspective. You cannot, look at one part of the cell and assume that if you hit it, nothing will happen. What will happen is that there'll be a cascading effect that messes up a lot of other things. So the whole idea of systems biology, coming back, is to make sure that we get some sort of handle on this complexity.
Okay.
Because this is a very complex system. And you don't want to do these spherical curve models. You want to make accurate models because each time you build a model, you're definitely making approximations. But the whole idea is that you want to have handles that you want to look at. Suppose I want to, manipulate the concentration of a drug and see what happens. Suppose I want to see what are the insulin dose I need to give to a particular patient. If you look at the standard models of drug action and so on, we have something known as a single compartment model. The body is a spherical cow, essentially. Right? The body is like one compartment where, all the, different, there's blood with some concentration of X met, various, whatever, sugar and whatnot and so on. And then you have this insulin coming in and it's going to cause some, you try to predict what is the curve, right? I put insulin here. There is a slight spike in glucose after that till insulin kicks in, and then there is a drop and whatever, right? So all these things are built using models, and models have today become so useful and so important that US FDA actually allows you to use models to file some of these claims and so on, right? So you can back up usually, of course, you have experimental trials and other evidences to back up your, drug applications and so on. But you can use models as a very legitimate component of your applications today.
Understood. So from what you said, a modeling of a system, of a biological system, would help me understand it better, make hypotheses on how to do different things, how to impact the system, and then maybe that's enough, or maybe then I would do clinical trials or experiments.
Absolutely. So let me just draw another parallel to engineering, because I think you've been talking about a lot of engineering aspects in your various podcasts. So
yeah.
Typical, what's your DBTL, right? Design, build, test, learn cycle. The same thing is there in biology as well, right? So in modeling, in fact, right? So you start with data or hypotheses, you design a model, then you run it, then you test. That's like the most exciting or scary part for most students and so on, right? So when you test it against data, you will find there are deviations. So now go debug, right? Why are these deviations happening? Why is, COVID, I'm not able to predict the spread simply because there are too many asymptomatic people. SIR model doesn't fit the bill now. I need to have another variable called asymptomatic, SIAR model or something like that. And I need to start factoring for these things to make my model more accurate. So design, build, test, and then learn from your mistakes. Learn from the deviations the model shows with respect to the reality.
Very nice. Thank you so much. Second question is, what is computational system biology? And also I've noticed that you've written a book on it. So I guess you're the right person to ask. What did the addition of the word computational change here?
Yeah, that's a good point, right? So systems biology has essentially two aspects to it, right? So one is experimental systems biology. Imagine, so how do you think one experimentally studies biological systems at scale? Which means you need these excellent methodologies to look into the cell. So I'll now throw a little bit more jargon at you, but hopefully everybody started hearing these, right? I think everybody started hearing genomics, right? So this omics idea is basically the om essentially is a sort of suffix that says that you're looking at the whole thing. Right. So versus looking at like one gene, if you look at all the genes in the human body, all the genes in a bacterium, you're talking about bacterial or human genomics. Or then, I keep telling my students, the most important aspect of biology you need to understand is just the flow of information in biology. Right. So there is DNA, DNA makes RNA, RNA is then translated to proteins, and proteins do all the hard work in the cell. Right. So if you look at all the DNA within the cell, that is called genomics. If you look at all the RNA within the cell, these are called RNA transcripts. Okay. And this is called transcriptomics. And then when you look at all the proteins in the cell, this is proteomics. Okay. There are excellent experimental methods that have been devised where you can study these things at scale. Or you can also look at what is known as metabolomics, which is like all compounds, all small molecules within the cell. So you can completely, characterize the cell at various levels, right? At the genomic level, at the transcriptomic level, at the proteomic level, at the metabolomic level, to understand what is happening within the cell, right?
Okay.
So, of course, I've not talked about the other thing, which is like microscopy, right? But microscopy is basically trying to look at the cell, look at what's happening within the cell. But again, there you'll end up focusing on like certain aspects of the cell, certain reactions or certain exciting areas within a cell and so on. But this is like a whole cell parts catalog in a sense. And systems biology or computational systems biology is about putting these parts together. We all have these slides and images which just shows like, a big box of Legos or like a big list of parts from an engine or something like that. Can you really look at that and divine what the original engine is or what the original system is? Truly not, right? So you need to look at the system, you need to look at the parts, and then you need to build the models that will try to answer these questions that connect the parts to the system.
The word computational here seems to imply that there's a lot of data computation that's happening. These data are coming from these experiments that you're talking about?
Yes. So these data form the basis for building the models. I now know what are all the metabolites that are present in a cell. What are all the compounds, small chemical molecules that are present within a cell. Can I now figure out what are all the reactions that are present? What are all the chemical reactions happening? Can I figure out what proteins are catalyzing these chemical reactions? And can I find out the rate of these reactions from the level of RNA within the cell? Because this RNA concentration will be proportional to protein concentration and so on. So essentially, there is like a whole network within the cell because all these things interacting with each other. Can you start figuring out what controls what impinges on what, right? So when I change this, so many other things are going to change downstream. So how do I figure these things very carefully?
So it becomes a correlation, causation, identification kind of.
True. Causation is like a real challenge here, but definitely we see lots of correlations and we use that to ultimately build these models.
Okay. So then because there's a huge availability of data, it then becomes a mathematical computation.
Exactly. Right. So to really, go back to your question. So systems biology, experimental systems biology is trying to do all these expensive experiments to characterize every molecule within the cell. Every DNA molecule, every protein molecule, every small molecule.
So like the genome project would be an experiment.
Exactly.
Okay.
Right. And we have the Genome India project, in fact, which we are a part of and so on. So we'll get to it when we get to it. But there are so many exciting things that are happening within the cell. And you need first experimental data, and then you need models on the other hand. And then we go through our DBTL cycle, right? So then using these data, we build models. And then these models, we use them to ask questions about the cell and then see, okay, this makes sense, this does not make sense. My model is predicting that this particular enzyme is not essential for cellular function, but when I remove it, the cell is dying. So what's happening? Go figure. Right? So this is our design, build, test, learn cycle.
Understood. And as you're explaining it, I get the sense that, I guess you said it in one of your lectures also, that this is a field which is very interdisciplinary. There's a computer science element, there is a biology element, there's a chemistry element. It's all coming together, right?
Yeah.
Professor, some words that you have used: biome, metabolite, and I think you used the word pathway also in the beginning. What do these mean?
So this is, just think of this as the, so biome again is, it's literally another word for an ecosystem, right? So all the biological stuff that resides in any environment, right? So typically you will see the word microbiome a lot more, because we are most interested about the microbes that reside in any environment.
Right.
Because, and I think it's more to do with the fact that we haven't seen them enough, or we haven't considered them enough, and we've suddenly realized that they are just so central to life and health and well-being and so on, right?
Yeah.
So you have like a ton of microbes on your skin,
Yeah.
right? So they have an impact on skin health and so on and so forth. And similarly, you have a lot of microbes that reside within the body. The original estimate was something about 10 times the number of human cells. But now the more accurate estimates are coming out as like 3 or 4 times the number of human cells. So I don't know, are we human or microbes, microbial, right? So we are outnumbered 3 to 1 or 4 to 1 by microbial cells.
And this is not just the gut, it's skin, gut.
This is everything. Yes. So there is this beautiful, I think, Science or Nature cover image which has a picture of a human, but almost, it's just microbes. Right? So it really tells you the story that that microbes are, and the other really cool thing is that there is another picture that you will see in all these review articles and so on, which ties microbes in various parts of the body to every single disease. Right? So you have the gut-brain axis. That is like talk about how the gut microbiome has a significant influence on autism and so on. So there is this whole set of things that are manipulated or influenced by the microbes in the body. And that is why there is this quest to understand them better. And if you see what we talked about systems biology, right? I would more narrowly classify the work I do as systems microbiology. Because we typically don't work enough with human systems. We have done some work, but we typically work within a single microbial cell, trying to understand the single microbial cell and, for instance, how it interacts with other microbes, right? Does it help or hinder the growth of other microbes and so on? Right?
So your work with microbiomes spans from, I've read, and maybe you can explain it a bit, spans from gut microbiome to microbiomes in the space station. Is that right?
Yes. And in fact, even further down, right? We've started with the deep sea. We've looked at some deep sea hydrothermal vents data. We have a fantastic collaborator in Wisconsin, Madison. And from him, we have these data sets from the Guaymas Basin of California. Right? And there you have these microbes that live under the sea. And there are these deep sea hydrothermal vents are essentially like deep sea volcanoes. And so they are very hot. And the sea, of course, is very cold. And what you have in the midst is the gradient of temperatures where various kinds of microbes survive. And typically, these are extreme conditions. So the microbes that survive there, we call them extremophiles. Right? They like these extreme conditions, or they tolerate these extreme conditions. And because of this, they have very exotic metabolisms. Right? They cannot have, like, your E. coli or your usually famous bugs cannot survive in those temperatures. So they need to have different pathways. And so these pathways are essentially, long chains of reactions. Some A becomes B, B becomes C, C becomes D, and so on. So, for instance, there is a big pathway through which the glucose that we ingest is converted to energy.
ADP
is produced. So glycolysis is one of the, classic pathways. And then you have what is known as the tricarboxylic acid cycle, and so many other pathways are there.
Okay.
So the very classic, so this is what a biochemist does. A biochemist tries to characterize all of these pathways. And I show these pictures in my talks, and you will see this on the walls of many a biochemist. There'll be like this huge poster which shows all the pathways that are happening within the human body. It's very intimidating. And if you still look at it closely, it's only like a very simplified view of what happens within the cell, because we only typically show only the metabolites in that picture. Where are the proteins? Where are the RNA? You can't ever.
What is metabolite?
Metabolites are these small molecules.
Okay.
So it's just a metabolite. So you can, in school we talk about them as compounds.
Okay.
Right? So metabolite is just a more technical name, because they are part of these these are actually called metabolic pathways. Because metabolism is basically breakdown and synthesis of various chemicals within the body, within a living cell.
Right. So my system's definition of a microbiome has to have the metabolites that are being measured. Is that correct?
Yep.
The
yep.
metabolites are being
So
measured, and what we're
typically,
trying
what
to
is
do?
measured is only the microbes.
Okay.
Right? Everything else is actually extrapolated. I wish we could measure the metabolites as well, and people are developing some of these things and so on, and people do that, right? So they do stool metabolome, right? Because you try to find out what are all the metabolites that are present in the gut and so on. But most classic microbiome experiment is you try to find out how much of each microbe is there.
Okay.
You literally try to create one vector of abundances. I have X microbe at Y abundance. Right? So X1 microbe at Y1 abundance, X2 microbe at Y2 abundance. I have this long vector.
Okay.
Right? So this is really the most important data that we work with. Right? So this vector will change based on deep sea, gut, International Space Station, or anything else in between, right? And how do these microbes interact with each other? So when I find high levels of X1, do I also find high levels of X20? And how do they interact with each other? So these are some of the questions that we like to
Professor,
ask.
immediately the question comes to my mind. There are three examples: gut microbiome, extremophiles, and the microbiomes in the ISS. It makes obvious sense to me to study the gut microbiome, because you're saying that gut microbiome can influence
health and disease.
Health in many ways. Obviously, daily health, but also maybe something like autism and diabetes, maybe. I don't know. Why are we studying extremophiles and microbiomes in ISS? Doesn't it sound like it sounds like an exotic problem?
That's true. So, but the thing is, if you see, there is a lot of push towards green manufacturing today. And green manufacturing in the context of how do you make, say, drugs or various other, or even like, say, butanol or ethanol, which is, a common blend in petrol today and so on. So how do you microbially synthesize these things? And for this, the typical quest is, can I find exotic microbes with pathways that can do this? And so there is this very classic
metabolite is a useful compound.
Metabolite is typically a useful compound, right? And there's this very classic, fantastic study, very innovative study, where there is this anti-malarial drug called artemisinin, right? That is only produced in certain plants. But what they did was they copy-pasted this pathway from plants to microbes to mass manufacture it. So this comes under the purview of metabolic engineering. This is again something that we really like doing in our lab. So we build a lot of models that can help in metabolic engineering, which is essentially how do I tune these microbial cells to overproduce a particular compound of commercial interest. So how do I, and it could be a single cell, it could be like a combination of cells, like a microbial co-culture. Can you use that because there is division of labor? Can you have one microbe help the other produce a particular metabolite or a compound, chemical compound, and so on? So this is the reason why we try to look at all kinds of, we like to go around characterizing organisms everywhere. Right? Can we characterize organisms in the IITM soil? Maybe there's like one microbe that is resistant to something and is some salt or something, and it's able to produce an exotic metabolite. And so much so, the example for this is, in fact, our collaborators have identified this compound called surfactant in the International Space Station microbes. And that compound is heavily used in cosmetics. So they have this very nice term of space to your face, right? So it's actually an organism that was identified in the International Space Station, but then that is able to produce this compound called surfactant, which is a very valuable molecule in cosmetics and so on.
Very interesting. There was a word you used. That's actually the last word on my list of terms I need to understand what you're saying, which is the word network. What is the context of network in the work we are talking about?
So literally, I mean, there are networks at various levels. Right? So in general, a network is simply, or a graph, as computer scientists call it, or mathematicians, is just a collection of nodes and edges. I think all of us are familiar with social networks.
Yeah.
Right. So in Facebook or Twitter, your nodes are people or businesses, and the edges represent connections and so on. Yeah. It is essentially the same thing, right? But here we are looking at the social network of a cell, or where the nodes could be microbes, and the connections could be the interactions. This microbe influences the other microbe and so on. Or you can delve within a cell there are networks at so many different levels, which is what makes it fascinating. And if you see, that is the most recurring theme in our lab. I don't think you will find we work on a various set of things. We work on metabolic engineering applications in one sense. We look at microbiome, we look at omics data analysis, we even look at systems theory where we collaborate with chemical engineers and so on. But across all of these domains, you will see that networks is a completely recurring theme. And this is because we study networks at various levels. So one is microbial networks, then get inside the microbe. Right? There you have these networks of various molecules, pathways. Every pathway is essentially a network, right? So there is glucose that is connected to another molecule it makes, that is connected to another molecule it can break down into, maybe 2-3 molecules it can break down into. Depending upon the catalyst, you might have different outcomes of different reactions. So you essentially take a large catalog of chemical reactions. This is, in fact, a very key aspect of most of our work. What I want is a large list of chemical reactions, A plus B giving C plus D. In fact, let's say 2A plus 3B giving C plus D. I like to know the stoichiometry, right? So the coefficients of these, how many moles, how many molecules are required to make this other molecule and so on. And using that, we have this long list of reactions. We then translate it to networks of different kinds. In one network, I would have metabolites as nodes, like all these chemical molecules as nodes, and what it can convert to as edges and so on. But then you can go further within this and still start looking at a single chemical as a network, right? So you now have carbon, nitrogen, oxygen as nodes, and all your double bonds, single bonds, chiral bonds as edges and so on. So in fact, this is actually, we've used this to predict how new molecules can be made. And now very recently, we've even used this to predict the synthesis of drug-like molecules and things like that.
Okay.
So I would just like to sort of, again, say that networks are just all pervading within the cell. And it's a very important aspect of computational thinking.
Yeah.
To really, you cast any problem into a network problem. And the standard, in fact, the other analogy I use is this essentially like Google Maps, right? So all of us have used Google Maps to get from one place to another. And you try to find the fastest way to get from one place to another. That is nothing but pathfinding on a network. And essentially, that is what we do in metabolic engineering, loosely speaking, right? I want to find the best way to get from molecule A to a commercially important molecule. And the problem is I need to reroute the traffic within the cell so that I can get more of my molecule of interest. So the cell is trying to grow. Cell is not trying to make ethanol for you. But as a metabolic engineer, I want to push the cell to make more ethanol. Right? So that is so the cell wants to do something. I want to push it towards something that's commercially useful in an industrial process.
Understood. Once you convert it to a network, all kinds of algorithms that are used in network thinking can come
Exactly.
in. Is this a word that you use often, molecule of interest? It sounds very quotable.
Yeah. Because we always have ethanol might be your molecule of interest, surfactant may be your molecule of interest, artemisinin may be your molecule of interest, right? So using these various, we want to engineer the cell. Metabolic engineering is all about rerouting pathways within the cell towards your molecule that is commercially viable, commercially important.
Very interesting. So I'm just going to sort of go through all the terms that I learned from you. Systems biology, computational systems biology, microbiome, metabolite, molecule of interest, pathways, modeling, and network in the context, extremophile, which I'm fascinated by the word extremophile. I'm going to just use it everywhere for people. Like, you can survive this, you're an extremophile.
Yes.
It's great. Thank you so much. I'm sorry this comes so late, like we've been talking for 20 minutes. But now I must ask you, you head the IBSE, right? Can you tell us what it is and what is the work that is actually done?
Yeah.
What is your research area?
So IBSE is, we started out as the Initiative for Biological Systems Engineering back in 2015 under the mentorship of Professor Ashok Venkitaraman, who was then at Cambridge. So he was like, we have lots of really bright young minds at IIT Madras who are very quantitatively inclined. Let us get them to work on biological problems. Let's not worry about generating biological data, which is like an expensive proposition. But there's so much data that's available online, which we can really leverage to do big things with it. And well, now we've gone as far as generating our own data, right? We are part of this very large national project called Genome India, where for the first time we've been involved in sequencing, and in fact IIT Madras has been involved in analyzing these sequenced genomes. So we have close to 10,000 Indian genomes that have been sequenced. And I think that's a great achievement, and it again gives us lots of insights into the kind of mutations that are present in the Indian population, what it means for our health, disease, and so on. Right? So going back, IBSE was set up in 2015 as the Initiative of Biological Systems Engineering, and then given how we've evolved, we've started calling ourselves a Center for Integrative Biology and Systems Medicine, because we want to focus on medicine, right? So IBSE, now the E stands for the last E in medicine, right? So it's like a contrived acronym. The thing is that so I head this center along with Professor Himanshu Sinha from the Department of Biotech, and Himanshu and I have been sort of leading this center from 2016, and it's been a fascinating journey where we have faculty from different departments. We have faculty from chemical engineering, from computer science. Now, of course, a few of them have moved to data science, including myself, and we have faculty, and of course biotech. So we have faculty from all these departments that come together to work on very exciting problems at the intersection of, say, biology, modeling, engineering, and so on. Right. So this is what we do, and we are involved in large, international and national projects. One of our biggest success stories is the project that Professor Himanshu led, which is called the Garbhini project, where this was a study on preterm birth in India. And it turns out that all the models that we have for gestational age are based on like American populations and so on. So they are really not ideally portable to the Indian context. So Himanshu and his students have developed new gestational age models for India and so on, which are much more accurate and things like that. So this has been like a major contribution from IBSE. And we are part of various other international initiatives, one of them being MetaSub, which is trying to look at, I'm going to throw some more jargon at you, which is metagenomics, right? So we've talked about genomics, which is the study of all the genes within a single cell. Metagenomics is a study of all the genes in a particular place. It could come from different organisms, right? So typically, if you're trying to study the gut microbiome, you're going to do gut metagenomics, because I need to take all like a stool sample or something like that, and then sequence it. And I will find a list of all microbes, and using that, I generate that vector I was talking to you about. How much of X1 is present, how much of X2 is present, and so on. So we are part of this MetaSub consortium. So MetaSub initially started off with, I think probably around 2015, where they sampled the New York subway. They just swabbed New York subway, right? Various surfaces, the handrails, the ticket kiosks, and so on, to see what is the microbial signature of New York City. And now we've done that for Chennai. So it's like a really exciting study that's currently under review. And what we find is Chennai has its own unique microbial signature. There are a few organisms that are present only in Chennai. And typically these come from the skin and so on. And interestingly and happily, we didn't find enough resistant organisms and things like that. So it's a very interesting signature. And we've also been studying other environments. For instance, we've been looking at some drinking water data sets. And we hope to also look at sewage water data sets and so on, because these can all track the flow of resistant microbes and things like that, right? So as we build towards smart cities and so on, genomic surveillance is also going to be important. Right, so you can see, the change in these microbial loads, and maybe they have a story to tell in terms of like a disease or like so on. So
I do vaguely remember reading about in the newspapers about the microbial load. I'm using the term of COVID in sewage water across cities and how it's changing, and that could be like a
definitely. Yeah. In fact, there was this, really, see, some of these are a little gross sounding because there's a lot of, work with stool samples and so on. But they actually analyze airline waste, right? To see what is the transmission of microbes across continents and things like that.
Very interesting.
Right? So all these, so we potentially have a chance to surveil these at some point of time, right? So this has still not become mainstream, but I would really imagine that in like a decade or two, this will definitely become mainstream, where we start surveilling microbes.
Gut microbiome work is pretty mainstream now, right? Like everybody is talking about it, and their
No, but
products hitting the
to use
market
it as
all
a
the time.
surveillance tool.
Oh, okay.
Right.
Okay.
So
Fair
definitely.
enough,
So we have so we do have the basic tools now, which have been really improved and fine tuned. But to actually regularly use it for surveillance, to make sure that we catch like an outbreak ahead of time and so on, I don't think we are there yet. But there are like tantalizing possibilities.
Professor, can you just some of the work that your group has done maybe is of common interest. So I was really fascinated by the work that is done with respect to the gut biome. There's a lot of conversation that happens online about gut being a second brain. I don't know what that means, but I assume that means that.
So in fact, I will say that rather than a second brain, it's probably the most important metabolic organ. Maybe you can, I don't know, probably the jury is out on to rank it above the liver or below the liver. But it's literally a very important metabolic organ because it produces a lot of useful metabolites for us. Various compounds that we are unable to synthesize on our own. So I'm sure you remember, you must have unfortunately taken antibiotics at some point of time, and your doctor would have likely prescribed like vitamin B alongside the antibiotics. Or in fact, now if you see a lot of doctors actually prescribe what is like a concoction of microbes along with your antibiotics, right? Because they know that by having antibiotics, you're going to like really upset the gut balance of microbes. And can you actually instead ingest certain helpful microbes that will go and restore the balance faster, right? Or worst case, you're not going to be able to produce some vitamin B in your gut because you've taken these antibiotics. Can you take a supplement of vitamin B so that you don't get blisters or whatever and so on, right? So this is I think so the gut is actually a very, very important metabolic organ because it does a lot of stuff. There's like a whole million, there are trillions of microbes there which basically produce a lot, and they break down a lot of compounds that humans aren't able to break down on their own, right? So whatever you eat gets broken down through the metabolic pathways and enter the gut. In the gut, these microbes and their metabolic pathways break down these molecules into much simpler molecules and so on. Many of them are very helpful for you. And then, there's also a lot of interest in what happens to these, the drugs that we take, right? So whatever medicines that we take, how do they get broken down in the gut? What kind of do they, what is the impact on the gut microbiome and so on? So all these are very interesting questions.
Just building on that, but maybe I take like one antibiotic course a year or maybe two because, well, that's how my metabolism is. But there are others who react to antibiotics very differently, right? So the impact of antibiotics on gut must be different for everyone.
Definitely, right? So potentially everyone's gut microbiome is unique, right? I mean, it's not like unique that probably you can group it into a few categories. So there was this classic paper that appeared maybe like 10 years ago, which said there are like 3 major types of guts and so on. But again, I don't think there was enough Indian representation in the data and things like that. So we may have to do this study. There is an Indian gut microbiome study that has been done, right? I think they've looked at about 3,000 Indian gut samples and so on. And we are still waiting some of the results from that study. And I believe it will be really fascinating when we get to understand what is the abundance of various microbes in the Indian guts, right? And we were actually part of a very exciting study with our collaborators in Singapore, where they actually studied what happens when you take antibiotics. And they had a nice cartoon to show that it's like a forest fire. So when you take an antibiotic, it's like a wipeout of what happens. It's not a true wipeout, but it really has a significant impact on the gut. And then slowly, as the forest ecosystem recovers after a forest fire, your gut nicely recovers after an antibiotic administration, right? But sometimes it's anywhere between 6 months to 2 years. And you're saying that, you might take another dose of antibiotics within that period. So your gut is, runs a serious threat of being dysbiosed, as they call it.
If it's unique for everyone, then that supplement that the doctor is giving has to be unique for everyone, right?
Well, very good point, right? So this is a very interesting question that we've been grappling with, and a lot of people have been saying that while the gut is, how do you say it? It's the organisms wise, it might be unique, but function wise, it's quite similar, right? So in my gut, there may be different organisms that produce the same lactic acid or, the compounds that are like the vitamin B2 or something that's required for our health, as in your gut. It might just be a different set of microbes that are doing it. But the point is, can you somehow push the gut towards those healthy things? And typically, this would be something like, can I improve the pH of the gut? Right? And so I'll give you these 3 microbes that will improve the pH of the gut. Something like that, right? So typically, we look at the production of various acids like propionate, butyrate, and so on, to just make sure that the gut health is improved and so on. But today, we really want to systematically design these concoctions, and for which again we believe modeling will be very handy.
Sure.
Right? I know that, hey, if I put these microbes, my gut will start improving, right? So these microbes will interact in different ways. So in fact, a lot of our work involves, of course, starting with a list of reactions, converting them into a network, and then I try to put these networks together and see how these networks interact with each other. Is this organism producing like a particular molecule that this organism the other organism can benefit from and grow faster? Do they collaborate? Do they cooperate? Right? Or is one being a parasite on the other? It is just, pushing out something to kill the other organism. So we try to study these things to understand which microbes can coexist, and ultimately which microbes can help each other grow or, restore gut health and so on.
Interesting. Although I must say that, I know that you're talking from an engineering point of view, but from the marketing point of view, it feels like there's some 10,000 new companies which are selling some gut-related product, and I have no idea whether they have gone through this rigorous modeling testing. I don't know what they're doing.
Yeah, so a lot of them, they probably, I'm not sure, some of them do some basic modeling and so on, but in general, they just look for, key helpful microbes that'll potentially produce some beneficial compounds, right? So most of these are harmless microbes, so I think it's not a problem if you ingest them, because microbes are just so much all around us. And I don't know, I think perhaps in school we are taught that microbes are bad, and, we actually use them as a synonym for germs, no?
Yeah.
And it couldn't be farther from the truth, right? Because you have far more helpful microbes in the body than, harmful microbes.
Understood. Damn cool. I do, as you are speaking, realize that maybe from a mechanical engineering, electrical engineering perspective, there's a lot of difference in the way you are addressing everything. It feels like in mechanical engineering, electrical engineering, we're building a system from scratch, from components, we understand. Here, you're trying to sort of break down and figure out how a much more complex system works. Is that a good understanding?
Yeah, that's true. That's true, right? So there's this famous article that I usually share in the first class in my course on systems biology. Right? This is called, Can a Biologist Fix a Radio? Right? So it really looks at how a radio is normally fixed, and then if a biologist were to fix it, how would he or she fix the radio? Because a biologist typically does not have an understanding of all the components within a radio, right? Or within a cell for that matter. The idea is an electrical engineer really understands, some electrical engineer really understands every component in the radio, and therefore he or she put it in. Right? Whereas if you look at a biological system, you have no understanding of how to. So if you open it up, again, a mess of wires, or you can't even see it, or if you see it in the microscope, you see a bunch of molecules running around, right? So what do? I go and pull out one capacitor or one protein from the cell. And what happens to the cell? Right? Typically survives. And then you're like, okay, so this protein is not useful for the cell.
Professor, I remember this article. So first you start with a sample of 10,000 non-working radios. And then from the first one, you pull out a capacitor. And see.
Yes. Right. So you literally have to, everything has to be done by reverse engineering. Right? Or like you're like sort of blindfolded with one hand tied behind your back, and you have to figure out this system, right? Because, and this is where modeling is actually very helpful. Because it is not very ethical to do some of these experiments on biological systems, right?
Correct.
Right?
Yeah.
You'll be killing you. Killing microbes is ethical, at least as far as we are concerned today.
At least it's not ethical.
Exactly, right? Whereas, if you're doing any experiment on any higher organism, there are so many ethical considerations, right? And these models can potentially help us answer these questions and create more meaningful hypotheses for us to evaluate in a lab and so on.
Nice. As you're saying it, I'm thinking there should be an Indian Institute of Reverse Engineering. Professor, this is fascinating. I'm very curious. You have such a grasp of the subject, and you're speaking at it with such speed, and I really have to catch up. But your journey till here started with a chemical engineering undergraduate, right?
Yep.
And then from chemical engineering undergraduate, you went to a
computational science. So in fact, my undergrad was in Institute of Chemical Technology, where there is a very fascinating course. It's a BTech in Chemical Technology, but you can specialize in various fields. You could specialize in textiles, you could specialize in polymers, you could specialize in food engineering and so on. And my specialization was actually in food engineering. And you can literally think of this as like the precursor to all the BTech biotech courses that we have. So what we studied was, we studied chemical reaction engineering, process control. In fact, process control is really what caught my imagination. And process control is literally no different from systems biology. Right? You basically start building models of reactors and how to control them. And here we build models of cells and try to figure out how to control them. So I would really think that my fascination for systems biology probably started in my undergrad. But then what helped me there was, we had a course on, or at least we used to have these something like what we could call today a 2-credit course, like half courses. So I had a half course on microbiology and a half course on biochemistry. And those things really gave me a perspective on all these molecules, and it gave me the ability to work with these things without being afraid of them, because I used to hate biology like everybody else in school, right? So now this...
But that's more a reflection of how biology is taught in
exactly.
school.
Or even what biology is taught, right? So both of these things are there, right? So what biology, even my son today tells me, hey, why should I draw all these diagrams and so on, right? So we still focus, so biology becomes like essentially a drawing exercise or something like that, right? So you typically still look at parts of a flower and things like that, which is a very important aspect of biology, but that's more traditional biology. Today, biology has become so quantitative and molecular. Right? You need to understand the molecules. In fact, in school, in our 12th standard chemistry, there was this very fascinating chapter on chemistry in action. And that talks about DNA and the genetic code, and how the genetic code is conserved across organisms. And from the genetic code, you can find out what is the protein being made and so on. It was really fascinating. Right? But we do less of that and more of taxonomy. Remember who was the scientist who discovered this particular disease or this particular plant or this particular...
Even there, some of the scientists who have discovered these drugs and plants have gone through a fairly computational process. They've done a lot of experiments. They've figured out so many things. And that part is just not mentioned, right?
Yep. This definitely requires some reform, right? I keep, because it's a problem for people like me, because I need students who like biology and like mathematics. And I have to typically tell people that, hey, don't worry about biology, you will start liking it. You will when I start teaching it, because
that's very interesting, because you mentioned that, and a lot of times students make a choice between biology and mathematics. It's an either
hate
or.
Biology, so I go for math. I can't do math, so I'll go into medicine or biology. So I think this has to change, because we've had any number of discussions about how to integrate, say, mathematics before, and today we are talking about AI into, say, medical curricula. It is important, right? And there's a very famous and controversial quote, I think, from the head of NIH or NSF, who said that biology is too important to be left to the biologists. And he said that the innumeracy of biologists is being a problem, right? It's being a stumbling block for development. Right?
As you're saying that, I'm thinking that we spoke of the COVID example, right? Like how COVID was spreading and all. That would be a fascinating chapter to study in school on how we...
Definitely. And it might be a very simple way to introduce trivial modeling in schools, right? Because all I want to do is if I'm able to predict from knowing x of t, I'm predicting x of t plus 1, right? I'm actually doing a prediction, and that's probably fascinating for a child, right? It's because as modelers, we get fascinated by simulations, right? Because we see, oh, I never expected this. Why is this happening? And you get to learn a lot by asking these what-if questions of various systems.
Nice. Thank you so much for that. I forgot how we got onto this. There's a note here that I have written which talks about genomic studies being like a very like a point in time in the evolution of biological engineering per se, and the Genome project, which was not the Indian Genome Project, but the Genome Project in the 90s. Was that something very is it a big step?
It's huge.
And
huge,
what was the impact
right?
of that?
I mean, it's both ways, right? So I think when scientists first started off with it, just like AI, right? So I don't know if you've seen the first AI. So when the first perceptron was invented, people said that they talked about AI advances that are not even conceivable today as mainstream by late 70s. They would be mainstream by the late 70s, right? So in similar fashion, when the genome project was conceived and when the genome project was nearing completion, people felt that, hey, we will solve disease. And it turns out, and that's where again, I will make a pitch for networks, is that there are very few single gene diseases. Every disease is actually a dysfunction of the complex network that is within the body. Which part of the network is failing? Which part of the network is malfunctioning? That is what we need to find out today. And you need a combination of methodologies from your omic technologies, right? Genomics, proteomics, or whatever, to models and so on to really figure out, hey, okay, this is what is not working, and how do we repair this? How do we fix this? What's the ideal way to fix it? Or even so in your gut, right? Your gut is dysbiosed, you have taken some medicines, or there's some problem, there's this inflammatory bowel disease or something like that. All these are highly correlated with the gut health. How do I give you the right set of microbes that will restore gut health? Right? So these are all very interesting, potentially engineering problems even, right? But the point is that you needed this kind of data to start looking inwards. Right? And what the genome project showed us was that, okay, there are only so many genes that are available. There are so many genes, but not all of them are associated with the disease, right? It is not so straightforward to map genes to diseases and so on. So this becomes very interesting, right? So how do you now start mapping genes to diseases?
But also a point in time where the amount of data available suddenly exploded, right?
Yeah. So that is because of the improvement in technologies, right? So if you see, there is this famous thing of Sanger beating Moore. Right, we all know about the Moore's law, which talks about the transistors in a chip doubling every 18 months and so on, and it's sort of kept pace for a long time. Might taper off, but Sanger is the one who he got 2 Nobel Prizes, and Sanger is the one who started this whole idea of sequencing. So till today, you do something called a Sanger sequencing to verify if your sequencing is correct and so on. So Sanger sequencing is a very basic sequencing methodology, but then you have various other things that have evolved today. So you hear a lot about what is known as next generation sequencing. Doesn't mean much, but NGS is the keyword that a lot of biologists were throwing around for the last decade or so on. Wherein you have a very interesting method of sequencing by synthesis. You have a gene, you synthesize a copy of that gene, and as this copy is being synthesized, you emit light, saying this is an A, this is a T, this is a C, this is a G, different components of the genome, the ATCG bases that make up your genome. And there is a sensor that captures that and says, hey, your genome is ATCG, GG, whatever. Right? So this became cheaper. And what you have is typically, when I see these plots, these are exponential on a log scale. Okay.
Understood.
So you see like a rapid increase, especially if you see the last 5 years. You probably generated as much data or more data than you generated in the previous 50 years.
And as experimental systems biology improves, that will just keep growing and growing and growing.
Exactly.
Fantastic. So, Professor, just to go back to that thread of your career, after your UG, you jumped to computer science PG.
So I was always fascinated with computing, and of course, towards the middle of my undergrad, really started getting drawn towards modeling and so on. So at that point of time, I wanted to really explore, how much more we can do in computing. And IISc had this perfect course for me, which was an MTech in computational science. And in fact, today we will call that an MTech in data science, right? And that course seemed like ideal because it had courses on high performance computing, on computational methods, and so on, with lots of electives. Strangely, my undergrad, as much as I enjoyed it, we didn't have the concept of an elective. And contrast that with IIT today, right? Yeah. Where we have 40% of our courses as electives, and I have my students taking all kinds of students from so many departments taking my course. So when I went to IISc, I had electives, and I took an elective on computational approaches to drug discovery, and a course on elements of structural biology. right? And all of these gave me very interesting insights into how we can sort of use computation across various domains. And I'm always someone who thought that you need, I mean, pure computer science is great, but I think you really have to apply it to a domain to really enjoy it. Right? You can still do the classic, just theoretical computer science and so on. But if you want to really apply computation, you should find an exciting domain to apply it. It could be transport, it could be mechanical engineering, it could be chemical engineering. There's just so many possibilities, or bio. And I felt that I had a big head start when it came to bio because of my knowledge of biochemistry, microbiology, and I was definitely interested in those quantitative aspects or like molecular biology and so on. So I thought this is what I wanted to do. And then I had like a very, very short conversation with my PhD advisor when we decided that we would start working on systems biology. Right. So I had, in fact, studied a lot about protein engineering, computational design of proteins and so on. And I went to her with a large write-up on that. And she said like, hey, but why don't we consider systems biology? And I was like, sure, right? Because I was always fascinated about modeling biological systems and sort of never looked back from there, I guess.
And how did you take a decision to become an academic, like a professor? Because from there you could have gone into industry, right? This is a field that a lot of industrial research.
Yeah, I mean, I was always fascinated by the academic world. So I think for the longest time, I remember I wanted to be an academic, I think it was sort of an easy decision for me, right? I remember when I was upgrading from my PhD, we had a lot of discussion, me and some of my friends. And at that time, I was like very clear, right? I need to do a PhD because, I need to, that's the kind of career I'm
That's the
looking
path
at.
to
That's the path to
professorship.
Yeah.
Is that the right word?
Yeah.
Professorship. Professor, okay. So now this is, thank you for sharing the body of work that you do. You're also a very popular professor on campus. Your courses are taken by lots of students. How has the relationship with students changed over the last 20 years since when you were a student to now? The faculty-student relationship.
Yeah. So I don't know, right? So I've been a faculty in I've been a student in different institutes. I've been a faculty only at this institute. And I think this institute has always been like a very student-friendly environment and so on. And even when I was an undergrad, I think we had faculty who were like really student-friendly and so on. And I think I tried to sort of imbibe the best aspects from these various places as as a faculty here, right? So I like to be a sort of combination of, strict yet friendly. Right. So I insist on my students to come on time, but, I give them attendance prizes and so on. This is something I've done from my first semester here. Right. So I've had a class once when about of 50 students, about 18 or 19 of them had 100% on time attendance. Okay. For the entire semester. Right. So I usually give them a book of their choice and so on. So this is, I think I've really enjoyed my journey at IITM, and it's very nice to be able to interact with students from various departments. And I really in fact, I've interacted with a couple of aerospace students. They're just so enthusiastic in my course on systems biology, and they come up with very innovative projects in the course and things like that. So I think that is probably one of the really cool aspects of being at IITM. Right. And I'm sure the students also enjoy that, where they sit in classes with students from so many departments. You do that in the first year, it's not a big deal. But when you do it in your later years, looking at cutting edge topics and so on, I think it's very fascinating because you really get very complementary perspectives and things like that. Especially
and the third year students by then also have a set of tools that they can bring. They have a perspective.
True.
First year students, of course, are new.
Yeah. They're just trying to stay afloat and, figure the system and things like that, right? And they still don't know their subject yet. Yeah. They're all school kids who are doing math sitting in the same class. So no matter if they are from different departments, it doesn't make a difference.
Professor, this was great. Thank you so much. It does look like this field requires a lot of patience. I'm curious to know though, what is the department or what is your research group doing in the next 2 to 5 years? What is it that you're looking forward to?
Yeah, so we are very excited about these various microbiomes that we have been studying, right? And what we want to really do is to make sure that we can understand the dynamics of these microbiomes. So we are still at this understanding phase, We eventually want to get to some, work closely with industry to build some products or what we call these concoctions that we've been talking, right, of that will help you restore your gut health and so on. They're called probiotics, at the risk of throwing more jargon, right? So these probiotics, how do we design them more systematically? Right? So can I design a specific probiotic for you?
Yeah.
Right. So because I know what allergies you have, or what is the antibiotic you're taking, or what is the disease that what is the infection that you currently have, and can we have the right concoction that will help you get out of this without much trouble? So, but for this, first we need to really understand understand the gut microbiome and the interactions that are happening there, and how do you sort of change the state of the microbiome? I have a microbiome in this state, and I want to push it to this state. So how do I push it? And there may be different paths, right? What is the best path? Right? So all these are fascinating questions that we like to ask and hopefully answer over the next few years. And we have a lot of tools that we've built over the years. So one of the things that we've kept on doing is that we've been building the various algorithms. So we have algorithms that, again, a lot of them or all of them use networks, but we have algorithms that tell you how microbes interact with each other. Right? How does one microbe promote the growth of the other, and so on. We have algorithms to design small communities of minimal communities of microbes that can have a specific function. And we have algorithms that can infer from various kinds of data sets. Right? So even in this, when you construct these metagenomic data sets where you have data sets from different you're trying to get the sequence of different organisms, you can get very little or like partial information or complete information, what is known as whole genome sequencing, or just like sequencing like a particular gene, which will give you, it's called 16S. It will give you only like a very specific insight into what kind of, it will only tell you what Escherichia is there, not that E. coli is there. I will not know the whole organism. So we have tools that we've developed to understand, to work with these data sets. So using a combination of all these tools, right, and building further, we want to understand the dynamics of microbial communities. I think that's very fascinating to us. For instance, are these communities prone to invasion? Like a new microbe, can it come and disrupt this, or it's going to get kicked out? Right. So all these questions we want to try and answer, ultimately, towards understanding how to manipulate gut health. Nice. Or manipulate the health of any environment. In fact, the other very fascinating study we've been doing is we've been studying corals in the Andaman, where we've been working with the National Institute of Ocean Technology. So they've done the diving to get the samples and so on. And we're trying to understand what is the microbial load present in these corals. And you see that in certain places, the corals are getting bleached and, dying out and so on. Whereas in other places, they are quite healthy. And we have collaborators in Saudi Arabia who've been working on the Red Sea corals and so on. They have seen that, they're able to restore the health of these corals by, giving a concoction of microbes.
Like right? A biological...
Probiotics for the coral probiotics.
Nice.
Right? So could we do the same thing here, right? So it's like lots of fascinating questions. And and typically I think of myself and our lab as the methods guys, right? So we build these methods, we are agnostic of what system we work on, right? It could be a gut microbiome, it could be an environmental microbiome, so on. But we try to build the basic math and the tools to build these models. And hopefully by using these models and tools, we'll be able to better answer many of these questions.
Nice. That's a great point to stop. Thank you so much, Professor. I learned a lot and I actually had a lot of trouble keeping up. But thank you for the patience and yeah, that's it.
Thanks. I too really enjoyed it. I know some of these can get a little technical, but I think, if you start looking beneath the jargon, biology is very easy to sort of navigate. And there are so many simple ways to introduce biology to like school children and so on. So you can definitely make sure that people can appreciate all the complexity that is there within a cell. Ultimately, these are networks, right? So the networks and the complexity, you look at all of them and finally start understanding how, and because we are always very interested in understanding a little bit more about ourselves, right? Because that's why the study of biology is the study of life. So it's quite fascinating to understand them at a network level and try to see how ultimately these insights can empower us to handle disease and health better.
Nice. Thank you so much. Okay, there you have it. If you have questions about what we spoke about, questions that I can answer or the professor can answer, drop them in the comments. And remember to like, share, subscribe. Thank you.